Thank you everyone who has joined. I just want to remind people that in lieu of a one-on-one meeting with Dr. Eichler, please feel free to join and participate after the seminar in a group discussion. So today, it is my great pleasure to introduce Howard Hughes Medical Institute investigator and National Academy of Sciences and Medicine member, Dr. Evan Eichler, as our seminar speaker. Dr. Eichler received his PhD from Baylor College of Medicine, where he worked on regulation, structure, and triplet repeats instability in the Fragile X gene, FMR1, under the mentorship of Dr. David Nelson. Dr. Eichler completed a postdoctoral fellowship at Lawrence Livermore National Laboratory before joining Case Western Reserve University in 1997 and the University of Washington in 2004, where he is now a professor in the Department of Genome Sciences and has been a Howard Hughes Medical Institute investigator since 2005. Dr. Eichler is also a member of the Brotman Beatty Institute and an associate member of the New York Genome Center, both of which are multi-institutional collaborations with a focus on advancing and exploring new opportunities in genomics and precision medicine for neurodevelopmental, neurodegenerative, and neuropsychiatric diseases and cancer. Dr. Eichler is a pioneer and leader in identifying and sequencing normal and disease-causing structural variation in the human genome. Indeed, his research provided the first genome-wide view of segmental duplications in human and primate genomes, and a major focus of his ongoing research is to understand the evolution and mechanisms of gene duplication and DNA transposition and their relationship to human diseases. As a testament to the impact and significance of Dr. Eichler's research, he has over 460 peer-reviewed publications with over 200 as first or corresponding author, an H-index of 176 and over 170,000 citations. Dr. Eichler has also made a significant impact on the training of future scientists as a mentor of 17 doctoral students, over 45 postdoctoral fellows, and over 25 undergraduates. Currently, Dr. Eichler serves as the principal investigator on 7 active grants from the NIH, Simons Foundation, and Brotman Beatty Institute that support his long-term research goals to accurately identify and quantify complex structural variation in human genomes and better understand pathogenic variation and its contribution to disease. So Dr. Eichler, thank you for taking time out of your busy schedule to share your research with us, and welcome.
Thank you. My goodness, that's probably the most comprehensive description I've ever heard. So great homework, Megan, in terms of getting it. Yeah, it's a treat to be here, and I'm going to share with you some published but some unpublished data regarding essentially the use of long-read sequencing, really to improve our understanding of human variation more generally, and new insights in terms of what we're learning about genomes and how they change, but also ramifications with respect to disease. So unfortunately, sometimes it freezes, so I have to reboot here just a second. There we go. So I like to start with this kind of very basic slide. Whether we work in mouse or Drosophila, or we're interested in cancer genomes, I think we all appreciate there's a wide spectrum of human genetic variation, ranging from single base pair changes all the way up to large chromosomal rearrangement events. And our ability to distinguish and identify these in patients and in samples has largely been technology driven. So in the old days, like 5 years ago, the way we would detect large scale events in genomes that would come into a clinic would be with cytogenetics and microarrays. The way we would detect single base pair changes in specific genes would be with sequencing. And as part of that effort, there's obviously this large zone of intermediate size variation, which is going to be the focus of my talk. We collectively call this structural variation as events that are greater than 50 base pairs in size. And you can think of these as insertions such as mobile elements, deletions, microsatellite, variable number tandem repeats, as well as larger scale genetic variation. And the reason I'm going to focus on this is that I'm going to make the case that, in fact, a lot of this variation has been missed, despite the effect of having genome sequence now routinely done for millions of individual genomes. So why is it so challenging? Well, the challenge is, and it's not just humans, this is true of pretty much every genome that we've ever looked at least every vertebrate genome, is that genomes have a complex organization of various degrees of repetitive DNA. So I'm showing you one form of complexity here. What I'm showing you is a representation of the chromosomes circa around 2004, 2005, where we're placing on this map all the large, highly identical duplicated sequences that are present more than 2 or more times in the human genome. So these are called segmental duplications, and I'm showing you a map of their location across within individual chromosomes. So wherever you see a joining line, that means there's a big chunk of sequence that's at least 20,000 base pairs and 95% identical to somewhere else in the genome. So about 4% of the genome is made up of duplications like this, or distributed between non-homologous chromosomes. So this might look like complete chaos, but it's not. It's actually highly organized. We have these interchromosomal, shown here in red, duplications between non-homologous chromosomes clustered near the ends of chromosomes, called the subtelomeric regions, or near the centromeres, which are indicated here by the purple, or a sequence that wasn't present in the human genome circa 2005. So why are those regions so important? Well, in essence, more than 50% of the copy number variation that exists between your mom and your dad's chromosomes maps precisely to these regions of the genome. And these regions of the genome carry about 500 to 800 genes, depending upon the annotation that you're looking at. So a lot of normal variation maps to these areas of the genome, but these regions have been particularly problematic in terms of sequence and assembly because of their large high identity. And this variation is extremely important in terms of both human evolution and in terms of disease. So this is something I'm not going to go into, but work from our lab and others over the last about decade now have shown that several of the genes, probably half of the genes important for building a bigger brain, especially the frontal cortical expansion, have to do with genes that are mapping precisely in those regions that have increased their copy in humans, but are not present in multiple copies in chimpanzee, for example. We also know that those duplicated sequences create instability in a genome through non-allelic homologous recombination, leading to common disorders known as Williams syndrome or Prader-Willi. And then there's a whole raft of different genetic diseases and genetic susceptibilities that is due to variation in these genes that typically cannot be characterized by simply applying deep short read sequencing to the characterization of patient samples. So for me, these dynamic areas of the genomes, these are areas that change very, very rapidly, that have not been ascertained by short read sequencing, are key to understanding both disease as well as the evolution of our own species. So what's the solution? Well, if you've been watching the literature over the last few years, you realize that there's a whole transition going on in terms of how we sequence. Long read sequencing is now becoming much more commonplace. This is work that we began, at least with other groups, about 5 to 6 years ago. But the idea that we can actually sequence genomes and sequence complex region has really become a reality, I would say, in the last few years. And I'm going to just give you a brief introduction of the two technologies that are dominating right now. One is from PacBio, so Pacific Biosciences. It involves single molecule real-time sequencing, where essentially you have these, what they call ZMWs, zero mode waveguides, which you just think of them as wells, little slits where a single molecule of DNA can enter. And then there's a tethered polymerase at the bottom, which incorporates in real time fluorescently labeled DNTPs. And you have lasers and optics that are actually picking up real-time incorporation of molecules that can range from 20 to 100 kilobases in terms of their size. The other technology is that of Oxford Nanopore, and I'm showing you here an example of an instrument that we got a few years back, a GridION. And the idea here is actually completely different technology. It's the idea, once again, of sequencing long DNA molecules, but the way this is done is by threading DNA through using an unwinding enzyme and a protein pore through a synthetic membrane. And as this molecule passes through, the single-stranded molecule passes through, it actually changes the current in very characteristic ways diagnostic of the sequence content such that that change in conductance can be used to actually infer the sequence. So these two technologies, very different in terms of their approaches, really have allowed us to begin to sequence, importantly, native DNA without cloning and single molecules in a really large, high throughput fashion. And in particular, and I want to emphasize these two, which have been critical to beginning to sequence genomes for the first time completely, have been advances really over the last 2 years. So PacBio has developed an approach which involves essentially high-fi or high-fidelity PacBio sequencing. And the way this is done is that you actually take DNA molecules, native DNA molecules, and you put hairpin adapters onto them. And so as the polymerase, think about it tethered to the bottom of that ZMW, starts to sequence, it actually loops around the hairpin and then sequences the other strand. And many of you have probably heard that the problem with essentially any long-read sequencing is very error-prone. But if you can sequence the same molecule multiple times, shown here is the molecule sequencing as you go around the hairpin multiple times, you can create sequences that are 20 kilobases in size that are about as accurate as the short-read sequencing data. So this is called hi-fi, or also known as circular consensus sequencing. This has really taken off over the last couple of years and allows us to generate for the first time highly accurate long-read sequencing. The big advance from Oxford Nanopore hasn't been in terms of the accuracy. It still is not as accurate as we would like it to be. But what it can do is it can generate what we call ultra long reads. So these are molecules that are over 100 kilobases in length. And now with a single Promethean flow cell, we can now generate close to 50 gigabases of sequence, where the average length of the molecule is over 100 kilobases in size, and sometimes molecules up to about a megabase or more in terms of their length. So this is incredibly important advance because it allows us to traverse, cut across complex regions of the genome so we can produce scaffolding in very complex areas. And I'm going to come back to both these points a little bit later on in the talk. So the first thing that you should know about long-read sequencing, it actually provides access to that structural variation at an unprecedented level. And this is true of both PacBio and Oxford Nanopore. I'm showing you data here from basically 3 families, a Yoruban family, a Puerto Rican family, and a Han Chinese family, where we sequenced an individual child with both short-read data as well as PacBio data and compared it to essentially optical mapping data. So this is using things such as bio-nanogenomics. So what this simple chart shows is essentially the gain. So the blue line represents the long-read sequencing with PacBio. The red lines represent essentially the sequencing that you would get with Illumina. And this is calling structural variants, or variants, I should say, from 1 base pair and up. So this would be a deletion to the left of 1 base pair all the way up to 1 megabase. And these are insertions from 1 base pair all the way up to a megabase. So the other thing I want you to point out is that you can see that the counts here are on a logarithmic scale. So while this difference that you see may not seem like a lot, on a logarithmic scale, we're talking about orders of magnitude difference. So the important thing, take home from this, is that when you sequence a genome, in this case, a human genome, and you sequence it with PacBio, you're discovering about 25,000 structural variation events. So this would be restricting to just the events 50 base pairs and greater. If you sequence the same genomes, the same 3 children, you would discover on average, and this is the best case scenario, about 11,000 structural variants. So there's about a 3 to 4-fold increase in terms of sensitivity of discovery of structural variation. In other words, 75% of structural variants that exist within a genome are missed if we simply sequence a human genome with Illumina. So when people tell you that we've sequenced your genome or we sequence it deeply, there's often implicit in this that means we've understood all the variation. And for structural variation, that's just simply not the case. So the long reads are like a human geneticist now has a different microscope with a different resolution. We can now see that intermediate variation that we couldn't see before, really for the first time in many cases. The other really cool thing about essentially the long read sequencing technology, it allowed us to finish work that we started over 20 years ago with the Human Genome Project and actually fully sequence and assemble a given human genome basically from telomere to telomere. So I want to tell you a little bit about this. I'm not going to go into great details. This is work primarily of Adam Philippi, who we collaborated with. But about a decade or more ago, 15 years ago, I suggested that we should actually think about finishing the genome by actually sequencing a genome that was actually a haploid equivalent. And so for those of you who are unfamiliar with complete heterotitoform moles, these are abnormalities of pregnancy where essentially the maternal complement of a contribution is lost. And so an egg essentially fertilizes, or a sperm is fertilizing an egg, the maternal complement is lost and the paternal complement becomes diploidized. So this grows into a mass and it doesn't make it that far during development. It's typically lost in the first trimester, but it produces essentially a haploid equivalent of a human genome, just representing the paternal, well, that paternal sperm that came in. So those cell lines do exist. And so we started years ago, started to characterize these as an approach to actually solving essentially the complete sequence of the human genome. So why did we need to do this? Well, in part because there's so much variation that exists between two haplotypes, i.e. your mom and your dad's complement. We wanted to simplify the problem by actually just having one complement to study. And so this became essentially the telomere to telomere sequence assembly that was being generated. And largely by applying both long-read sequencing technologies, both Oxford Nanopore as well as PacBio, we went from a genome that looked like this circa 2019, where we actually have, where I'm showing you here the current reference human genome, where white represents gaps, i.e. no sequence, and black and gray represent transitions where we have incomplete sequence, so not telomere to telomere or contig breaks. We went from a genome that went like this to applying the long read sequencing to a genome that at the beginning of 2019, when we started, looked something like this. By the summer of 2020 was almost complete except for the rDNA clusters located on the acrocentric chromosomes, to 6 months ago looking like this. So some of you may have heard of the finishing of the human genome. This was the effort that was done largely by applying long read sequencing with some algorithmic developments along the way. So what did we learn when we sequenced the genome that we didn't know before? Well, this may not sound very satisfying, but we recognized the genome is actually more complex than we thought. There's in fact more duplicated sequences. So instead of 4 to 5%, there's about 7% of it. So this is the same graphic that I showed you before, adding essentially a lot more duplicated sequence and complex repeats. But importantly, having the complete sequence now allows us to ask questions regarding the structure, function, and evolution of these new regions of the genome. So while there's only 182 new protein encoding genes that were added as a result, we now have a complete reference to begin to ask really fundamental questions about very complex regions. So here's an example of a region that was fully sequence resolved. This is an area of actually incredible disease relevance and also very interesting population genetically. This is an area of our genome that's about 5 megabases in size. I'm showing you on the flank here the organization of duplicated sequences. So these are duplicated sequences themselves that are 0.5 megabase plus in size. Each of those segments carries about 12 genes in it. Now, these are genes important in terms of intrinsic immunity. They're called defensin genes. And what we've been able to determine, here is the reference sequence, which looked like it was complete in the old human reference genome. But you'll notice two big differences. One is that there are 3 copies of this cassette of 600 kilobases. So 6 times 12, 72 genes versus essentially 2 times 12, which is 20, I guess 24, 36, I should say. And the other thing that you'll see is that the whole region is flipped around. So this region is a 4.11 megabase inverted region that in the human reference is in a direct orientation. It turns out that this inversion is seen in about 40% of humans that have been characterized. And variation of this defensin cluster goes all the way up to about, I think the highest number we have observed is 10 units in some people. So some people have close to 120 distinct, or highly related defensin genes, while others have only 24 or 12 such genes. The presence of this inversion actually predisposes to disease, particularly a congenital abnormality, because it leads to unequal crossing over in weird ways, creating essentially a deletion syndrome associated with 8p21. But this is just one example of what we now estimate as close to 50 regions of the genome that are highly dynamic and structurally variable between modern or contemporary humans. Most of that variation was really not sequence resolved, or is still work in progress in terms of sequence resolution. We also learned something about regions that we had never sequenced before. So for those of you who are unfamiliar, the short arms of chromosomes 13, 14, 15, 21, and 22 are called the acrocentric short arms. And these are like these little satellite regions at the end of our chromosomes. They were never sequenced as part of the Human Genome Project. And applying long-read sequencing, we were able to sequence them for the first time. Most people thought that those regions were composed of rDNA. So what is rDNA? rDNA are the ribosomal RNA molecules that provide scaffolding for the ribosomal complexes that are required for protein synthesis. And it's true, we can identify the rDNA clusters that are shown on each of these. These are made up of tandem arrays of rDNA molecules. People knew that these regions were also composed of satellite sequences, which are indicated here. So this is this off gray that I'm showing here. But remarkably, more of the sequence is composed of duplicated sequences, largely of unknown functions, some which potentially encode new transcripts. And this is what's indicated here with all these colored bars that I'm indicating. So almost more than half of the acrocentric chromosomes are made up of duplicated sequences themselves that have nothing to do with rDNA and nothing to do with satellite. And if you look carefully, I've color-coded them based on their kind of their ancestral origin. You can see that certain acrocentrics actually carry large blocks of similar sequences, which is consistent with what people believe is that these acrocentrics are capable of undergoing non-homologous exchange events, both in evolution, but also among human beings today. But here's the important point. Now that we have a framework and we know the organization of one set of acrocentrics, we can do experiments. So for example, we can take a probe now that we know maps precisely to this sequence, and we can then fish map it, for example, against normal humans. So here's an example of 3 different humans. This is the Hydatidiform, so this is a haploid human, and these are 2 1,000 genome projects, taking a probe from the acrocentric, which is completely conserved in all chromosome 13, 14, and 15. So this is proof that that particular segment is shared and appears to be invariable among humans. However, this is now taking a different region from the acrocentric, doing the exact same experiment. So on this Hydatidiform, it maps to chromosome 13 and 14 acrocentric, but also maps to chromosome 18 paracentromeric region. In this human, it maps essentially to chromosome 13, but only one of the 2 homologs of chromosome 14, and only one of the 2 homologs of chromosome 18. And in this human, it actually maps only to 1 homolog in chromosome 13, only 1 homolog in chromosome 14, and it's not observed in chromosome 18. This may not seem normal to you, but this is an example of what has been described in the literature as chromosomal heteromorphisms, where some chromosomes, depending upon who you are, carry different content of sequence compared to others. And so the fact that we actually have this very dynamic region, we can now assay it in this kind of very systematic way and know exactly where we're mapping, I think is absolutely revolutionary. For those of you interested in disease, chromosome 13, 14, for example, actively participate in Robertsonian translocation. And Robertsonian translocations are an abnormality, in fact, the most common abnormality of birth in terms of the birth defect. And then we now can, in fact, map the breakpoints in principle by actually having this fully established reference. So that's fine and great, but that's actually sequencing 1 haploid human. And to be sure, that's not sequencing the diploid human. That is applying long-read sequencing to kind of cheating in a way and sequencing one where we don't have to worry about having variation. So the next challenge, which is something that both the computational and the genomics field has taken on, is how do we generate fully phased and assembled diploid genomes going forward? And so I'm not going to go in great detail in terms of the different methods that are out there. There are now, I think, 3 or 4 distinct methods, but they all involve some kind of formulation or difference of this basically schematic. So typically what happens now when people want to sequence and assemble a genome, they generate high-fi, so this high fidelity PacBio sequencing, which allows you to actually build a pretty contiguous assembly already without knowing anything more. But they add additional information. That additional information comes from either having parent-child information. So if you were going to sequence a genome, you would sequence the DNA of a child and you would have the parental information. Or you can generate physical linking information in the form of high-C or strand-seq data from that same individual if you don't actually have parental information. And the idea is pretty straightforward. And this is just one formulation work that from our lab and Tobias Marshall is you would take essentially this hi-fi PacBio sequence data, create what we call a squashed assembly, identify all the SNPs that you can from that data, and then use, in this case, we use strand-seq data, but you could do the same thing with high-C data to actually physically phase those SNPs. So you knew which ones are potentially coming from one haplotype versus another. Then you would go back using those phase variants, actually partition your high fidelity reads into haplotype A or B, say paternal or maternal, and then assemble essentially two genomes as opposed to one. So you're using essentially either the parent-child tree information, the strand-seq data, or the high-C data to actually phase the SNPs and assign the reads and actually to build essentially a representation of both the paternal and maternal haplotype. So we did this as a proof of concept on one sample. This was a Puerto Rican sample. We did it without using parent-child information. So we used just that proximity data that we had from, in this case, strand-seq. And we were able to build a 6 gigabase genome assembly. This is kind of the end of last year, where the average contig N50, so the average size of the contig was 25 megabases in size. And so what I'm showing you here is haplotype 1 and haplotype 2. So instead of actually making one representation of chromosome 1, we now make two representations of chromosome 1 based on essentially that phasing data. And then we went back and said, well, did we get it right? So we went back then to the parental information, both in this case, so we had the mother and the father of this. And wherever you see continuous blue, that means we nailed it. We got it all right. We assigned all the chromosomal contigs from the paternal to the paternal haplotype 1. In this case, haplotype 2 was the maternal. Wherever you see a switch, where you sometimes have a blue intermixed with the pink, that means we messed up. We didn't get that one quite right, so that's a switch error. And whenever you see nothing, that means we couldn't make the determination, we couldn't assemble it, at least correctly, and phase it. But here's the take home. 97% of the genome we got right. Switch error was less than 1%. And the parts that we couldn't involve the centromeres and the acrocentrics and some of the largest segmental duplications. But I think this is absolutely, to me, amazing because we can now physically phase and assemble a human genome. Instead of thinking about it as a 3 gigabase thing, we're now thinking about it as a 6 gigabase, which means that all variation is put into its context with respect to its haplotype and whether it's structural variant or SNPs, they're all together. And so having achieved that, we decided to go back to the 1,000 Genomes Project. And this is the work of a consortium that I co-chair called the Human Genome Structural Variation Consortium. And basically to sample at least one individual from each of the populations from the 1,000 Genomes Project. So we managed to select 32 individuals, applied PacBio sequencing, and applied strand-seq, and built physically phased assembled genomes for the first time from a population sample. So this is an example of the kind of information that we gathered. So once we actually had haplotype 1 and haplotype 2, and we generated high quality data, either by error correction or use of hi-fi data, we got complete maps of SNPs. And so we know which SNPs are both, in this case, that are found in the paternal and maternal complement for these regions of the genome. And this is just showing you the counts in a window across each of these. And so areas like HLA, which we know are very diverse, pop up immediately here. On top of that, we get the structural variation. So for each genome that we're sequencing, we get the structural variation events. And this is just showing you the counts in those same windows. And this is a property that we identified a few years back We know that structural variation, about 45% of it maps to the ends of human chromosomes within the last 500 kilobases to a megabase of those chromosomes, but not all chromosomes, which we don't completely understand why. But having said that, 55% of the structural variation maps throughout. The really cool thing is that we have the ability now to put all this information together. We have the structural variants and we have the SNPs. So we know the haplotypes in which the structural variants actually arise. And so we can do cool things. And this is some of the things that we've been doing. So we've taken those 32 reference diploids and we've made 64 human genome references. We've computed all the structural variation events that are greater than 50 base pairs in size. So this is the cumulative addition by adding one essentially haplotype phased genome after another. You can see that Africans actually have more genetic variation. You get more bang for your buck, as predicted. 20% more structural variants are discovered if you sequence your next sample as an African versus a non-African. We realized that there are regions of our genome, about 1,800 of them, where nobody agrees with the current reference. So that means these are all structural variants where the reference is probably wrong. And then there's another roughly 5,000 sites or more, which in fact most humans have, which are shown here in purple, but in fact the human reference is the minor allele. And then we have polymorphic variants, which are shown here by the dark purple, and then the singletons indicated here by the light blue. But the really powerful thing is, and I really won't have a lot of time to go into this, is that once you sequence resolve, and once you have the flanking SNPs that are associated with structural variation, you can now go back to Illumina data and you can genotype those variants much more accurately in Illumina whole genome sequencing data. And so this is an example of taking sites that we identified and genotyping them back in populations from the 1,000 Genomes Project where we had Illumina WGS data. And we can show that something on the order of conservatively 50% of the structural variants, even though they can't necessarily be discovered in Illumina data, once discovered in sequence resolved, we can actually genotype them back in Illumina data. And we can do that by actually having fully phased information in addition to the actual breakpoints for structural variants. So a fellow by the name of Tobias Marshall has developed a graph-based approach to do that type of genotyping precisely. So our prediction is, and we have some early days, some positive examples, but I think there's much more to be discovered. There'll be new associations with disease and disease consequences because of the ability to now go after this missing variation much more systematically. So I want to share with you one interesting insight that we've made, and this is very recent data over the last, really the last 6 months or so. When we started building these phased genome assemblies of humans, which are indicated here along the bottom, we started comparing them with non-human primates, which are indicated here as chimp, orangutan, and gorilla and bonobo. We started to look at different classes of variation and how they compared between different species, particularly closely related species. And I'm showing you here one class of variation, which I haven't discussed, which is inversions. So anything in a direct orientation, like the reference is shown here in this blue, anything that's an inversion orientation is shown here in this orange. And inversions are often mediated by segmental duplications, which are indicated along in yellow, because the presence of large, highly identical duplicated sequences predisposes to unequal crossing over, leading to a flip in order of given sequence. And one of the things we realized about 1.5 years ago is that when we started to look at regions of the genome, that there are areas that seem to be toggling back and forth between the direct and inverted orientation. So if you look, there's a region right here, for example, flanked by these large, highly identical duplications, which is sometimes in a direct orientation, sometimes in an inverted orientation, but even in humans can be either a direct or an inverted orientation. And that's because those duplications have lived for millions of years at the periphery, at least that's the logic, and they're constantly flipping this region back and forth in terms of its orientation. But now that we actually have phased human genomes, and we know where the inversions are, and we know where the SNPs are, we can ask this question within the context of humans. And so this is a study from David Perubsky in my lab, but we went back and looked at essentially all the inversions that he had identified, and we looked at the flanking SNPs. And I think he had something like close to 200 such inversions that were large that he identified. And about 2/3 or more of the inversions show topologies like this. When you build the actual coalescent tree, so you can think of this as a genealogy of the inversions belong to essentially one clade, suggesting that they arose once during human evolution. And everybody who shares this inversion shares common ancestry, at least for this locus, from an initial inversion event. So the inversion event is indicated here in blue. However, there's about 1/3 of the inversions which show topologies that look something like this, where essentially these are the haplotype, the leaves here represent the different haplotypes on the human tree. And everywhere you see a red, that means it's inverted. When there isn't a red, it's not inverted. And we can infer, based on essentially the topology of this tree, that at least 15 inversion events must have occurred back and forth over time to create the actual haplotypes that exist in the human population. In other words, this form of mutation, inverting a segment of DNA backwards and forwards, has occurring at an extremely high rate of recurrence. So I think this is incredibly important because, in fact, if the inversion is disrupting a gene or disrupting a regulatory element, you wouldn't necessarily be able to impute it just by knowing the individual flanking SNPs. You actually have to know the inversion status, or you have to build a deep enough tree to actually see that. So the final thing, which I think is actually super cool, is if you look at regions of the genome that actually are predisposed to unequal crossing over, microdeletion and microduplication. So about something like 7% of all developmental disability and autism in the human population is due to unequal crossing over between duplicated sequences. We find that these areas of the genome that are toggling, inverting back and forth from a forward and direct orientation in normal individuals, there's a 5-fold enrichment based compared to a null model from microdeletions and microduplications to occur over those exact same sites. In other words, recurrent mutations and hotspots of recurrent mutation are actually predisposing to microdeletion and microduplication syndromes associated with things like Williams syndrome, Prader-Willi, Smith-Magenis, and so on. So we think this property of a genome is actually incredibly important for predisposition to disease. All right, so what I want to talk to you now about is actually going after some of those regions that we couldn't resolve in essentially those phased genome assemblies. So I told you that we could get about 97% of the genome, 96, somewhere in that range. So we've been interested, at least one postdoc in my group has been extremely interested in going after human centromeres. And for those of you who are unfamiliar with centromeres, they're basically in humans made up of 171 base pair motif that is actually repeated into a higher order structure, often of 6 to a dozen of these units, which are then repeated up to millions of base pairs in size across the genome. So these are areas of the genome that are highly repetitive. And the reason that they haven't been sequenced and assembled in the past is because you don't know really where you are in short or even moderately range sequence to begin to actually construct an assembly, because it's essentially wall-to-wall tandem repeat sequences over and over again. So Glenys Logston, when she was working on the telomere to telomere project, came up with this idea. It was pretty straightforward in essence. She goes, instead of trying to focus just on one technology or the other, in terms of finishing the genome, why don't we apply both Oxford Nanopore and high fidelity sequence and leverage the strengths of the two technologies? So the approach was take a human genome, generate ultra long ONT sequence data from that genome, take that same genome, generate high fidelity. So these would be reads of 20 kilobases in size versus the ultra long, which would be hundreds of kilobases in size. Take those high fidelity reads, build essentially your first pass of contigs with the high fidelity. You can use the phased based approach for that purpose. Once you've built those contigs, identify unique k-mers. So we call them SUNCs, for singly unique k-mers, that occur once and only once in the human genome. You can think of them as SNPs. They could also be prologous sequence variants between duplicated sequences. And then use those unique k-mers to barcode your ultra long reads that are hundreds of kilobases in size. And so once you've done that, you can go to these areas of the genome that you couldn't close and scaffold across the contigs with essentially the ultra long reads. And remember, with Oxford Nanopore, you still have errors. And so those scaffolds are only 97 or 87 to 98% accurate. But then go back to those very regions, now that you have them scaffolded and anchored to the left and to the right, and fill in with high fidelity directed sequencing. So really, this is local sequence assembly of the region. So Glennis did this for the first human autosome centromere, chromosome 8. So she publishes this in the summer. And this is showing you the organization of the chromosome 8 centromere. So just to orientate yourself, the P side, or the short arm, is to the left, the Q side is to the right. All these colors represent different classes of repeats, but the alpha satellite repeat, which is repeated over and over again, is just shown here as gray. But what we're depicting below is a visualization tool that was developed by Mitchell Folger, which we find quite helpful. It's called, essentially, he called it stained glass, and it's now available in bioRxiv, and he has a GitHub for it. And what it does is because it's nothing but a tandem repeat repeated over and over again, what this tool does is it basically chunks the segments into 5 kilobase pieces, and it compares every 5 kilobase piece with respect to each other. So in areas where you see bright red, that means those pieces of 5 kilobases are nearly 100% identical. But as you move away, let's say down into the blues and the purples here, that means that these pieces are more divergent. So they would be see at 70 to 80%. But what's beautiful about this is that you're looking at like a dot matrix here, is you can actually see the pattern because it's the same sequence repeated over and over again. It's just different degrees of sequence identity. You can actually see the pattern of relationships among that, both the monomeric satellite as well as the higher order repeat structures of the alpha satellite. So when they did this and they showed me the first views of this, to me it was actually striking because this is actually theory that was proposed in the 1970s by George Smith, who suggested that the way centromeric satellites evolve is essentially like an assembly line procedure where you have younger elements in the center, which are highly homogeneous, and as they move out, they become more divergent over time, such that your older elements be located at the periphery, and your younger elements be located in the center. So this was like classic textbook of what a centromere should look like based on the evolution of higher order centromeric satellites. The thing that was striking to us was that when we started to look at this and study it in more detail, we could see clear evidence of layers. So for example, this older, more divergent stuff is actually more similar to itself on either side than anything else. So everything else is less than 80 to 78% divergent here. And so you can see here, there's another pocket of sequence. It's not huge, it's 100 kilobases in size, but it's actually more identical to itself and on either side, suggesting two things. We have a layered organization in terms of the actual satellite sequences. And there's also a mirror symmetry that appears to exist, so that you can see that as you have in the center, you have this highly identical satellite sequences that is virtually 100%. You get this area that's more divergent, but then you get these pockets suggesting that there have been replacements of probably the active centromere over time to result in this structure. At least that's one interpretation of essentially the evolution of centromeric satellite. The other really cool thing you can do, and remember when Glennis was doing this, she was doing both using high fidelity sequence from PacBio, but she was also using Oxford Nanopore data. And Oxford Nanopore data has an incredible power to detect methylated CpGs. So it's one of those sequencing platforms where you can actually get the methylation status of your CpGs with pretty standard approaches now in the field. And so what she could do is she could actually build a methylation map across the centromere as well. And anywhere you see red, that means that the CpG is essentially hypermethylated. Anywhere you see blue, that means the CpG is unmethylated. And what we would expect for an area like a centromere, which is actually highly closed chromatin, we would expect most of it to be hypermethylated, which is the case. You can see that most of the centromere here is hypermethylated. But the really cool thing was this little dip in methylation. It's a hypomethylation dip, or an area which other people have described as CDRs, but it's an area of about 76 kilobases in size, in which there seems to be a paucity of methylation. So for those of you who know your centromeres, you know that it's critical to mitosis and meiosis. And we know that essentially the way that chromosome segregates, there has to be a kinetochore assembly complex that binds. And so the question that Glennis had was, is this the location of the kinetochore? So she did what would be an obvious experiment to her, but not necessarily me. She did essentially SEMP-A chip sequencing. And SEMP-A is essentially a protein that is diagnostic for the position of the kinetochore assembly. And so shown here is that same map I showed you before with the hypomethylation dip. And then shown here is ChIP-seq with SEMP-A, showing you the binding of where the highest signal is. And I think it's pretty striking, even though the region of SEMP-A binding is broader than the methylation dip itself, it seems to center almost perfectly over that hypomethylation region. And just to confirm this for reviewers, she went and actually then did fiber-seq where she stained with 5-methyl-C, and you can see there's a paucity of methylated bases here, experimentally determined, and then co-stained with SEMP-A, which shows that, again, it corresponds to the same place. So super cool, right? This is a new property of centromeres that really wasn't realized. Closed chromatin for most of it, but a hypomethylation dip located in some portion of the higher order sequence, which we now know exactly where that maps in this particular centromere, which defines essentially the epigenetic, the fundamental kinetochore assembly site, which we couldn't live without, right, for mitosis and meiosis. That is the power of long-read sequencing. So the other thing I asked her to do was actually, since she had developed this method, to go ahead and do multiple non-human apes. If you ever work in my lab, you don't just study humans, you study everything in an evolutionary context. And so she built out the sequence and assembly of the chimpanzee, actually both haplotypes. This is actually the haplotype organization. Again, you can see the stained glass view here once again. Interestingly, the chimp is similar in terms of its mirror symmetry, but you can see that it actually has what appears to be more structure than what we see in humans. It's a smaller centromere. It's about a megabase smaller, but it's essentially has more higher order and mirror symmetry in it than what we've seen before. Here's sequencing of an ape known as the orangutan. This is about 12 million years diverged compared to the chimp, which is 6 million years diverged. Once again, you can see this pattern of higher order structure, or I should not call it higher order, but layered organization. So for example, this particular divergent sequence here is more similar to the sequence on the Hughes side than it is anywhere else. Same for this segment, and same for this segment. But what's really cool about the orangutan, we don't know quite what to make of it, is that it actually has two areas of higher order structure that are actually highly identical to one another. And these two sequences do not appear to be that closely related to each other. So they're not just simple derivatives of each other, but they're in fact always as if there's actually two different higher order structures that may be competing for dominance of essentially centromere segregation. At least that's a potential hypothesis. Keep in mind that this centromere is actually much larger than what we saw in either human or chimp. It's 4 megabases in size. And then this is going back 25 million years, at least in principle, in terms of ancestral reconstruction. This is the macaque genome, diverged 25 million years ago from humans. It's massive. Its centromere is close to 11 megabases in size. So it's about 5 times bigger than the chimp in terms of satellite sequences. But its structure is actually quite simple. It actually has just one giant zone of almost perfect identical dimer motifs. It does show that divergent structure and layered organization, but not as structured as what we saw in chimp, human, or even orangutan. So once you actually have the sequence from the chromosome 8, and you know exactly that it's chromosome 8 syntentic, even though it's rapidly evolved between all 4 lineages, you can basically look at this sequence, extract the monomers, and actually build a phylogenetic tree. And so this is a simple phylogenetic tree derived from positional information that came from the full sequence assembly of these different apes and humans. And what this data shows, it shows basically an unrooted phylogenetic tree, suggesting that there are 2 trajectories for the evolution of centromeres. One that goes toward essentially the higher order structures that we see in the great apes. So green, black, and blue represent the ape sequences. The orange represent the macaque, but this higher order sequence is related to sequences on a very specific portion of the macaque centromere. So we can think of that as the vestigial portion in the monomeric or at the periphery, that blue sequence I was showing before, that gave rise to the common ancestral sequence that led to essentially ape and human centromeres. The flip side is also true, which I think is super cool. You can see the monomeric or dimeric structures of the macaque basically clustering over here. So this is the bulk of that 10 megabases that I showed you before. And it's related to a very specific portion of monomeric satellite located on the Q side of chromosome 8. So this is trajectory 2, leading to old world monkeys, and this is trajectory 1, leading to the evolution of centromeric satellites. So for any students caring about the evolution of repeat sequences, including the most complex repeats in our genome, we now have the ability to reconstruct even rapidly evolving regions and reconstruct the phylogeny of them. So what about human-human comparisons? So how much variation is there in a chromosome 8 if you actually sequence 2 human haplotypes? So this is going back to that Puerto Rican sample that I showed you in the very beginning. So we now have finished the chromosome 8 centromere of that Puerto Rican sample. This is the paternal centromere 8. This is the maternal centromere 8. You can see that the pattern is very similar. You can see the mosaic, or not the mosaic, you can see the layered structure and basically the presence of this symmetry. But if you look carefully, you can see that the zones differ in size. So this region, which we labeled as layer 5, which is the highly identical piece, is much larger in the maternal centromere 8. And this layer 3, which is this peripheral segment that is located basically at the edges of the higher order, is much smaller in the maternal complement. So we've been excited about this extent of variation because it is actually truly extraordinary. When you think about sequencing, let's say, a human haplotype anywhere else in the human genome, you expect a difference between your mom and your dad's DNA of 1 change every 2,000 base pairs. But in these regions of the genome, the amount of variation that exists between your maternal and paternal complements are on an order of magnitude higher. So this is aligning essentially the paternal centromere 8 centromere to the actual CHM13 reference. And then this is comparing paternal and maternal. And anywhere you see a straight red line, that means that's what you'd expect for normal DNA, 99.9% plus identical. Anywhere where you see blue or you see yellow means that it's much more divergent. And what you can see is that there's been so much change that the line is no longer a straight line. It actually blows up. So amount of variation that exists is absolutely extraordinary. So how does that look if we went back and redid the entire experiment over again in a second hydatidiform? So remember, the first telomere to telomere reference was done out of 1 hydatidiform. So we've been working on a second hydatidiform mole called CHM1. And now I'm showing you the amount of large scale variation at the megabase scale for 5 human chromosomes and their centromeres that have been fully sequence resolved for the first time. So to me, this, as a geneticist, this is absolutely wild. Some chromosomes look very, very similar to one another. So here's 2 different chromosome X's, and you can see the stained glass view is virtually identical. There's a little bit of a difference, but it's more or less consistent. But take a look at this chromosome 21. This is almost a 5-fold difference in size based on the centromeric satellite region that's present. This chromosome 17 is very similar, although this layer seems to be gone completely, right, in this particular centromere. But look at this chromosome 8. This chromosome 8 looks very different from the other 3 chromosome 8s that I showed you in terms of the centromere. It has a very large, expanded, highly identical 100% region. Or take a look at this chromosome 10. It looks like it's been completely dominated by an expansion of a new type of satellite that has taken over the majority of chromosome 10. So what we're doing right now is we're building a complete, kind of our second complete human genome, CHM1. And we're also comparing these systematically against chimps and other non-human apes to build a global framework for the evolution of centromeric satellite DNA. Coming back to this epigenetic question, because we have Oxford Nanopore technology, we can also ask the question, where is the hypomethylation dip in these different centromere structures that we see? And so if your eye can follow me for a second, if you go to the chromosome 1 here that I'm showing, there's a little hypomethylation dip that you can see. I can turn on my annotation here. There's a little hypomethylation dip located here, and it's roughly in the same location in the second chromosome 1 that has been sequenced and assembled. But take a look at chromosome 20. The hypomethylation dip is located roughly around position 2 in this schematic. But when you go to the second hydatidiform mole, you can see it's actually shifted almost an entire megabase away, suggesting that the actual epigenetic or the site or location of the kinetochore can change probably in concert with the sequence changes that are occurring, or maybe independent from it. But we can actually simultaneously map both the actual sequence data and sequence structure to essentially the actual epigenetic signatures. Oh, I think I have a freeze here. Oh, there we go. So this is just to highlight the change in that hypomethylation dip on chromosome 20 between the first two chromosome 20 sequence centromeres. So I want to end with the clinical genetics application in the last few minutes and say where this is going. I know some of you may have read the Danny Miller's paper, and I'll touch on this very briefly. But obviously, the ability to actually sequence these very complex regions of the genome is opening up essentially discovery of new mutations associated with disease. And so this is an example kind of a positive control that we did when we first started this. This is a child that has Barrett-Scott syndrome. And for those of you unfamiliar with Barrett-Scott, it's a particularly difficult disease or genetic disease to discover because it's due to actual triplet repeat expansions that occur on both copies of a gene called XYLT1 in the 5' UTR of the promoter. And so what happens in these particular kids is that the CGG repeat expands, much like Fragile X, leads to hypermethylation of the promoter and extinguishes the expression of XYLT1. Interestingly, it's an autosomal recessive disorder. Most of these diseases are autosomal dominant. And in this case, you need to actually have both alleles to be hyperexpanded. So this is the child shown here in black. It actually has a hyperexpanded, as detected by Southern blot, of an expanded allele, where the CGG repeat is growing to hundreds of units or thousands of units in length. So this is a mom and a dad that actually one was carrying an event but it was because it had two chromosomes that didn't actually have Barrett-Scott. This was the mom who had a premutation allele which expanded to lead to essentially this child with essentially this defect. So this is high fidelity PacBio sequencing of the mom to the left and the child on the right. And this is just to show that we can sequence resolve the fully expanded CG repeat and distinguish both alleles based on high fidelity sequencing. So nothing new, other than the fact that we didn't have to run a Southern and a restriction enzyme blot on this. We just discovered and we visualized the sequence and discovered that. But if you have Oxford Nanopore technology, you have methylation on top of that. So this is now looking at the proband and looking at the reads with the methylated islands shown here in red compared to the unmethylated CpG islands shown here in blue and the mom and the dad. And I think you can clearly see the child has essentially methylated most of its copies, which is why the child has developed Barrett-Scott. But what's really cool is you can see that there are two reads in the mom that actually look hypermethylated. And the data suggests this corresponds to this premutation allele, which is beginning to get larger, but hasn't gotten to a point that it becomes a target for almost complete hypermethylation. So I'm showing you this example because this is simultaneous detection of structural variation, complex sequence that could not be sequenced, and I'll say this with an exclamation point, could not be sequenced with Illumina, where we not only sequence resolve, but we also get the methylation status. So what Danny's been doing is been going back to all these unsolved cases, of which there are legions of unsolved Mendelian cases. And I want to be absolutely clear here. This is a little bit, I always remind him of this. This is a bit like shooting ducks in the, ducks in a barrel, I think is the analogy. Because you have evidence, you have a clinical diagnosis in terms of the phenotype, and you have a good guess which genes are affected. So this is not whole genome. This is going back to the specific genes of interest. And so there's a method in Oxford Nanopore called adaptive sampling, where you can do targeted long-read sequencing computational, so no CRISPR-Cas required, by just actually programming which regions of the genome you want to sequence. Of course, you have to have a complete sequence of the locus you want to study, but that's no longer an issue with the complete sequence and assembly of the human genome. And so what Danny has been showing, and he continues to show, is that he can solve about 25 to 30% of the cases of this type. So here's an example of a child that has Lesch-Nyhan syndrome. This is a neurobehavioral deficit due to mutation on the X. Nothing could be found initially. Danny sequenced with long reads, targeted long read sequencing, found essentially this deletion as well as a complex inversion, which affects splicing of HPRT. And again, subsequently shows that HPRT is not spliced correctly in this child. Here's an example of a child that has Alström syndrome. The point mutation was known, but as a recessive trait, no one could find the second allele. Danny found a deletion, or I should say in this case, an insertion, precisely in exon 20, disrupting the other copy. So here the power is targeted long read sequencing discovers missing, often structural variation, and also provides phasing information simultaneously. So you basically can actually really truly identify compound heads. And then the last thing, and this is probably relevant to people interested in cancer as well. This is not a cancer patient. This is, believe it or not, a child that has developmental disability. Sometimes in cases of developmental disorders, there's a complex series of rearrangements that occur, which is often referred to as chromothripsis in the field. So there's like basically, you can think of the part of the genome essentially explodes and then reassembles itself. So Danny did targeted long read sequence of a very large region across multiple chromosomes based on FISH results that came from that patient. This child obviously had lots of chromosomal abnormalities, including translocations and other types of events. So people knew that that was probably the cause, right, of why this child had developmental disability. But in most cases, either by array or by cyto, you don't have breakpoint resolution. And so what Danny did is he said, let's just target these entire swaths, about 100 megabases of sequence across these different regions. And when he did that, he could identify about 12 breakpoints. And this is what this subway plot, as he describes it, is showing you. It's actually showing you the transitions from one chromosomal region or one chromosome to another, precise long reads that are connecting one part of the genome to another based on long read sequencing, targeted long read sequencing of this patient. Why is it important? Well, when he had breakpoint resolution, he could identify essentially 12 genic breakpoints, so things that affect genes, of which 11 were previously unknown. And 2 of them look like they may be relevant to the disease etiology of this patient. So that's just the increased sensitivity and power that you have from the long reads to really go in and give precise information on the breakpoints. And this is why I believe that long read sequencing is the future, not just of basic research in human genetics, but of clinical research. I don't think in 10, 20 years from now, we'll be talking about Illumina, at least in this way, Illumina sequencing. All right, and so in summary, I hope I've convinced you that long read sequencing just rocks. It just absolutely rocks. And it rocks because it's providing us access to essentially structural variation, most of which has been missed previously. So it's like we have a new microscope in our field, right, for discovery of both normal as well as pathogenic variation. Combining hi-fi sequence data or linking read technologies such as TransSeq or Hi-seq allows us to actually physically phage genomes and start thinking about a 6 gigabase genome as opposed to a 3 gigabase genome, which I think is incredibly powerful for both studies of disease, evolution, and population genetics. And telomere to telomere assemblies of genomes are possible. And I think the solution will come from routinely combining both Oxford Nanopore's scaffolding potential as well as the high fidelity accuracy. So far, it's only been demonstrated for haploid equivalent genomes. So the next big challenge will be showing that we can generate a diploid telomere to telomere assembly. We can do specific regions now that we never could do before, but no one's demonstrated it for a complete diploid. Then lastly, we're providing new insights into the complexity of human genetic variation. Think of the inversions and the recurrence of the inversions. And in terms of disease, it's going to lead to new discoveries, new mechanisms of disease, I believe. The impact of rare variances will become even more obvious, I think, to people as we do more of this type of work. But the idea that we're actually comprehensively understanding human genetic variation is just fundamental to essentially, I think, not just humans, but to all research involving genetics. I'll just put a plug, and I won't talk much about this. Some of you have heard, I think some people at Tennessee are involved. I'm involved in this, one of the PIs of an initiative now to not do this just on 30 samples, but now to move forward to 350 human genomes. And so look for these papers, hopefully, God willing, in 2022. We're beginning to actually sequence an even better representation of the human species. And I think I'll skip this, but just simply to say there's incredible areas of diversity out there. Some of it we're starting to see with graph-based approaches, and then we're linearizing this. So this is just one locus, a gene family that is thought to be important in terms of building a bigger brain. And remarkably, humans, and these are just examples of human haplotypes, differ radically. So every little red line here that I'm showing represents an individual gene copy. And so there's the heterozygosity among humans in this locus for this gene family is close to like 90%. Our data suggests that there are probably 50 to 100 such sites that contain genes that are highly variable between humans today. Most of that variation hasn't been systematically studied yet. And the graphs, which I'm showing you to the left, is one way to access that. And I'm sure Eric will be happy to tell you much more about this at some point. So these are the folks that did all the work. I want to specifically call out Danny Miller, Glennis Logston, and Mitchell Folger, students and postdocs in my lab that have actually driven this research. For the telomere to telomere work, I don't want to take credit for that completely. That's Adam Philippi, Karen Miga, but in particular, the two Serges, Serge Koren and Sergey Nurk, who actually took advantage of the long read sequencing that data that was generated to really close some of the remaining gaps to create the first complete human genome. And then for the genotyping, which I didn't spend a lot of time on, but also for the phage genome assembly, work from Tobias Marshall and Peter Ebert at Dusseldorf and Saarbrücken in Germany. And with that, I am happy to take questions. Thanks.
Most excellent.
Hey, Rob.
I will defer questions because I have too many, but let some other people ask.
I guess I can ask a question to get people started. So I had not seen the centromere work before, and that's really interesting. So what is the significance of those differences in structure and hypomethylation? And I mean, does that potentially cause problems with, I mean, what is the significance of that in terms of like disease or like structural variation?
Yeah, I mean, I think one potential significance, right, is we know that aneuploidies, right, in the human genome are due to precocious segregation, right, of chromosomes. And when you actually have, and I mean, this is theory now, but you know, people have talked about a model of concerted evolution of centromeres. And so satellite sequences aren't necessary and sufficient to have segregation. But when you actually have a satellite sequence, it seems to reinforce maintenance of a centromere for that location. So we know that neocentromeres can evolve in new locations of the genome, and that it's usually that the satellite sequences evolve secondarily. So the ability to actually, you know, constrict and actually have a kinetochore assembly component bind is usually happens first, and then the actual satellite sequence evolves second. And the satellites actually compete with each other to who can be the most competent. At least that's the theory out there. So centromeres are constantly evolving kind of in an arms race with each other to basically segregate themselves efficiently and appropriately when mitosis and meiosis is occurring. So what I think we're looking at, I mean, I think that incredible dynamic change that you're seeing in terms of the centromere organization over such short periods, we're talking hundreds of thousands of years, is a reflection of that arms race and concerted evolution. And so different satellite sequences are expanding and contracting to become better at in terms of recruitment of the kinetochore for actually driving segregation. And so obviously we have to have many more centromere sequenced and assembled to maybe make rhyme or reason out of it. We have some ideas on what we've seen because we can now correlate those dips with the actual sequence itself. And there's an old theory out there called the library hypothesis, which says that you have all these different flavors of satellite sequence, in this case, alpha satellite. But the most competent satellite is often formed by taking different satellites and bringing them together. And so that you don't actually just have a homogeneous sequence. And that's one of the things that we've learned is that the most homogeneous region of every centromere doesn't necessarily correspond to the site of the epigenetic signal that corresponds to the kinetochore assembly, but it's the diversity of satellites that exist at this location that seem to be driving it. So you can think of it as like a mix and match. It's always like trying different satellite sequences to say, I want, this one is a little bit more efficient than this one, because obviously if you have a more efficient centromere, then you're better off at actually undergoing segregation. If you have an inefficient centromere, then you are more likely to have precocious segregation and aneuploidy, and that makes you a dud. So it's actually cellular selection, right, as a consequence of segregation occurring probably as a consequence of this interplay between the epigenetic and genetic signatures. This will probably not be the focus of my lab going forward. I've been brainwashed by Glenys Logston, who's about to launch her own lab. But in many ways, this goes back to work I did many years ago on tandem repeat sequences. So it's been a lot of fun to work with her on this and apply the tools that we have to these areas.
Yeah, it is really mind-boggling to see all that hidden stuff in the genome and think about like what it might be, what it might be doing or using or what's going on in evolution. So that is actually really, I'm always amazed when I see these talks.
Yeah, and you know, from an engineering point of view, and even an evolutionary point of view, it's quite surprising to me. You'd think that if any part of the chromosome was under intense selection, as you said, even at the cellular level, it would be that mitotic apparatus. It's really it should be under really tight control, stabilizing selection, and you wouldn't expect it. It's almost paradoxical, counterintuitive that you would pile in so much variation into part of the chromosome that's so critical for every cell.
Unless the dynamism is key to the evolution of the structure, right?
Yeah, I think exactly. And that's telling us it's not an engineering issue. It's trying to be really flexible and strong in the face of God knows what.
I mean, for us, just a completely different perspective outside of the centromere. Now, for us, one of the greatest kind of realizations is in the duplicated regions of the genome that are predisposing to disease, because these are also very unstable regions, right? They're constantly changing. The reason that we have Williams syndrome or we have Prader-Willi is because duplicated sequences have evolved over the last 10 million years to create a lability in our genome at this position because of unequal crossing over. But that maybe not the irony, but the remarkable thing is that these very same regions also carry within them the duplicated genes that are important for building a bigger brain. So like RGAP11B or SIRGAP2C and all these genes, they are in the duplicated territory. So without this additional dynamism, we wouldn't have actually evolved to actually build that bigger brain. But the consequence of actually having that is essentially increased susceptibility every time we produce germ cells of having a microdeletion or microduplication predisposing our kids to autism and developmental disability. Yeah. So it's like there's an overall selection, right? It's a cost of being human, right? In a way, just like there's a cost of actually, segregating chromosomes, right? In our genome. Yeah. And in a genome like ours, where to be honest, from a genetics perspective, we are slow evolving, right? Our mutation rate, it was been documented for decades, is extremely slow. Most of the genome is kind of snail pace, right? Chimp and human, only 35 million single nucleotide differences between our two genomes, 6 million years. In Drosophila, it would be huge, right? But in us, it's like a spit in the bucket. having this kind of extra source of diversity, I think, is absolutely key to us being a plastic organism in terms of being able to respond to evolutionary pressures.
I would like to follow up on that, but let me be quiet for a minute and make sure everybody else gets a chance to ask questions.
So there is a question in the chat about junk DNA.
We don't call it junk anymore.
That's politically incorrect now.
Well, no, I mean, that's what people, when I was
No, I'm just kidding. I'm just kidding.
No, I was a student when they did definitely refer to this as junk DNA. What's the question? I'm sorry, I can't read it. It's actually.
Oh, it says the human genome has a small amount of junk DNA of unknown significance. With your long read sequencing, have you been able to identify or map junk DNA in quotes to regulatory regions or structural variants?
Actually, better than that. We've been able to map that junk DNA to brand new genes that do different things, specifically in humans compared to chimps. But yes, to all of those things: regulatory DNA, changes in structure of the genome, changes in gene content in the genome, all of that. And in terms of the proportion, depending upon how you do the math, I mean, most people think about half of our genome is made up of junk. So it's not a small proportion. It's actually quite a large proportion of it. And to be fair, a lot of it doesn't appear to be doing anything. But you know, it's finding those diamonds in the rough, right? There are now dozens of genes that have evolved very recently as a result of that. And they hit above their weight in terms of their impact, right? What we don't know is, because people haven't looked at variation in those genes, is how variable they are among humans, which is what we're trying to do right now with the HPRC and HGSVC projects.
I'm just going to paste in some questions for you, Evan, and maybe also ask them. The first one, I just wanted to come back to the first one is the global one. Could you please rewrite? Oh, sorry, I've got Michael Lynch's book, The Origin of Genome Architecture here. He wrote it in 2008. I would love to have you rewrite it. It's a great book. It was an eye opener for me, but 10 years is a long time, or 12 years, 13 years. Somebody like you has to rewrite it. And it's not going to be Mike Lynch, I don't think, because he's pretty preoccupied with other things. But it'd be good for you. There's the famous last chapter in this book on the everything is adaptation argument, which he goes on a total flame. It's pretty damn funny, if you're not the target of it, that is. But it would be wonderful to have an update, because your presentation is so exciting. But I worry that everybody doing genetics that I know of in the animal model work, they're still thinking forward genetics. Start with the phenotype, drill down, do it over again. And they'll be doing it for 100 years. And really, these sequencing technologies just flip everything on its head. You should just take your population, sequence them all, and see what you can do using reverse genetics with common variants.
Yeah, no, essentially, I mean, how we built the lab, right? Years ago, we used the duplication architecture to predict where the Cnvs would be that cause disease. So we went, and then we searched for phenotype, right? So our lab, we always joke, is definitely a child of the genome project, because we started with the genome sequence first and moved out, right? And that's how we discovered genes involved in the evolution of the human brain as well, and not with a phenotypic first approach. It's a genotype first approach going forward. But Rob, I'll promise you, when I get closer to retirement, I'll consider it. I just I've had lots of discussions. I did, and I haven't recently, but a decade ago with Michael Lynch. And I always would argue with him that, I appreciate all the work he's done in terms of the field of duplication, but he always tried to create an average of like duplication rates and stuff across many, many different taxa. And as we start to delve into individual, whether it's Drosophila or mouse or humans, we see all these idiosyncrasies. And he would always say, oh, that's just, it all averages out in the end. But I said, maybe all of evolution in terms of the dynamics of genomes, maybe it's all idiosyncratic. Maybe every lineage has their own path. And I said, averages mean almost nothing in that context. I mean, averages mean something if there's a normal distribution, right? And we approximate the norm. But when we don't, I think this is where the long read sequences is really going to tell us this, right? Because people are applying the long read sequencing, whether it's to many strains of yeast, natural and inbred, or lab strains, I should say, or mice or plants. People are starting to see what the pattern of variation looks like much more comprehensively. And I think once we establish those, there maybe will be some general rules, but I think in some respects, you know, looking at short read and incomplete assemblies, which is what we've done for 2 decades, really is like putting both your arm and one leg, right, behind your back, so to speak. And you know, you just don't have enough information. And so I think this is so beautiful now that, you know, you can make a complete genome assembly pretty much of any vertebrate you want as a grad student. You know, it's the idea of doing that. That was a, you know, $1 billion endeavor with, you know, 30 labs 2 decades ago. Yep. And now we're talking about tens of thousands of dollars to do it well, right? Which is amazes me. Right?
Yeah. Megan, do you want to unshare Evan's screen so we can get everybody?
I can stop share here.
Channel? Yeah, there we go. And then we can get everybody on a gallery mode. All 15 of us who are left.
Hey, Evan. This is James. Thank you so much for this nice presentation. I have a very naive question, actually. I mean, you know, actually, there's a variability between the human beings to human being. But what do you think within our body? I mean, do you think there's kind of variability within the cells, especially for the brain? This is my first question. And second question, you also showed the hypermethylation or hypomethylation. And that also, I mean, from this perspective, actually, in the organ, I mean, from one organ to other organ, I mean, this kind of methylation might be different, because you show one results for hypomethylation. I mean, is it coming from one which tissue, or could it be different from the tissue to tissue? Especially for the brain, can we see that kind of variations?
Yeah, so those are good questions. So the first question has to do with somatic variation. We haven't done a lot on this yet, but others have. So I'm thinking specifically people like John Moran in terms of MEI, Jeff Faulkner, and other people. And it's clear there is variation that occurs between individual cell and cell lineages somatically. What I will say, and I think this is also borne out by cancer as well, is that when you look at structural changes that occur germline versus essentially what you see in those, somatically, it's not the same rules. So, for example, when you look at structural changes between humans that occur as a result of germline mutations, a large fraction of the big events are driven by non-allelic homologous recombination events between repetitive sequences. That does not appear to be the case for somatic mutations, at least associated with cancer, or even the somatic events that have been seen in normals. The problem, and people are working on this, but problem with long-read sequencing is it's not terribly amenable to single cell approaches yet, and that's because large input DNA sequence is required. Or you have to amplify, and the minute you amplify, you take away some of the power, right? Which is essentially, real-time native DNA, whether it's methylation or structural variation detection. So that's a limitation of the technology, is that it's not ready for single cell, although there are people, and I know some people working on trying to develop that. Yeah, so that's the somatic question. And your other question, I'm trying to remember, what was it?
Hypomethylation, hypermethylation.
Oh yeah, so a question of what types of cells. So what we've been looking at so far, just so you know, is lymphoblastoid, which means EBV transformed, and fibroblast. But for example, the CHMs, which I showed you that one slide, those are fibroblasts that are basically being compared. And so that's fibroblast to fibroblast comparison for whatever it's worth in that case. But you're right. I mean, an epigenetic signature could change in principle, right, even between cell types. And I think it's a great question. It would be nice to have that kind of data. And one could imagine generating it now from, the cooperative tissue human networks, right, where we actually have autopsy material from multiple tissues from given donors, right? That would be, I think, a very fascinating thing to investigate, whether the epigenetic signature stays the same,
Yeah, I
right,
mean,
in the same
in
location,
the
given a
centromere.
genome has, I mean, the other question is, which we often puzzle about, is, now this hydatidiform mole is, a haploid human, so to speak, and it's been passaged, but how much mutation has occurred in passaging in the structure of the centromere, for that matter? How stable is this thing?
Yeah.
I don't know. I mean, there is data from pulse field that's 30 years old that suggests that it should be more or less stable, because multiple pulse fields from the same individual gave the same result, but that's not the same thing, right? So it's those are all great questions, which can be answered now, for sure.
Thanks.
Evan, what is the relationship between recombination hotspots and cold spots and the new higher level of large-scale variation you're detecting? Can a region of high variation be essentially a sink for recombinations to keep haplotypes intact?
Yeah, could be, because I mean, I don't know how many of you know about normal recombination, but normal recombination in males is biased toward the ends of chromosomes. So in females, recombination is much more uniformly distributed across the chromosome. And so this has been known, work from Terry Hasselt and others. I think it's the same in mouse too, but maybe not to the same extent. But there's this huge bias, at least in male germline, for recombination events to occur in the last last tens of megabases of human chromosomes. It's kind of ironic if you remember, I showed you that very brief map of structural variation. I said, 45% of structural variation maps at the ends of chromosomes. It turns out that the chromosomes that show some of the highest rates also show the highest rate of double-strand breaks. So, which kind of makes sense, right? More double-strand breaks, more structural variation. But the flip side of that is that means that any two human haplotypes must have greater diversity at the ends of the chromosome because of that. And as a result, recombinations should be suppressed. Yet it's the opposite way, at least in paternal, right? Male germline. So yeah, I think it's kind of a conundrum a little bit. If we have this, it must not be enough differences to disrupt recombination activity, right? Because one idea would be that if you had enough structural variation, you would stabilize and prevent recombination events from occurring at the edges anymore. But it's clearly not the case in males.
Is anybody in your group working on that issue of how it all syncs up with recombination in the two sexes? No,
not right now. I mean, there was a postdoc a few years back, Beth Dumont, who went over to the Jackson lab that was very interested in this question. I think she's still interested in it from the perspective of both normal recombination as well as gene conversion.
Yeah. I would love to understand. I have not heard a plausible explanation as to why most rodent, or at least mouse and rat, chromosomes are telomeric, and in human we have what, 5 or if you include Y.
Yeah, the telocentric. Yeah, I don't know either. It's kind of one of those. The simplistic argument is the amount of rDNA that needs to be produced as a function of the metabolism of cells and cell division, right? And so, the simplistic view is if you have a high metabolism organism, which I would put mouse in, right? They need to go through many more cell divisions, and therefore they need to have a lot more rDNA and more ribosomes to basically keep up with the high demands of a fast living.
I've heard that.
Yeah, I've read it somewhere. Yeah,
I wonder if are telocentric.
Yeah, but it's true that, I mean, genomes have huge variability in terms of both the copy number and the number of acro or telocentric chromosomes, right?
How are we doing on the Y chromosome? The CHMs are XX, right?
Yep. So we've gone back and as of, I think, a month ago, Adam Philippi was able to assemble there's a second human reference, it's called HG002, which is the Ashkenazim from I don't know if anybody knows, but this is a trio that's been set as a standard to try to build references. And he was able to fully sequence and assemble the Y. least that's was his claim, and I believe Adam. But he was like very frustrated by it because the last bit, which I think was the satellite sequences on the Y, were incredibly painful.
Yeah, it's
That's
kind
like
of funny.
something-
How many final versions of the human genome are we going to have, Evan? About 6?
Well, now we have one for sure, right? We have essentially the heterotitiform mole, CHM13, and the agreement is to just add the Y from HC02 to that one. So that would be then X with and the Y, right? And then I think the next challenge will be, well, it should be, I mean, we're going to try to work on the CHM1, so that would be a second. But then there's the push to do the first diploid, which will be an effort of the HPRC kind of going forward. But, you know, in the end, we won't be doing reference-based genomes, right? I mean, again, this is a separate talk, but the idea, like from Eric, right, is the idea that we would build from those a graph-based reference, which would continually improve dynamically over time as we sequence more and more completely sequenced human genomes.
Drunk that Kool-Aid for sure. I didn't quite understand so you described in quite a high level of detail the phasing problem, and the solution and the accuracy of your phasing is 97% plus accurate. When I look at my 23andMe, which is just based on lousy, 500,000 SNPs, they do give it to me with phase. And is that basically fantasy phase, or how good do you consider that?
That's real phase. That's real phase. But remember, there's two different issues here. One is phasing a genome, which we could do with SNPs, and I mean, for a while, right? All we need is to know who's het and home across the entire genome. So that's you can consider this as superficial phasing, because that just tells you the most common variants, either your het, home, and then you can partition to haplotype 1 or 2. But what you really want is you want to assay all variants, irrespective of frequency. So whether a variant is seen only in the Williams grandparents or in the Williams ancestor that lived 5,000 years ago, there's a big difference there. And so this is where imputation comes in, because people try to guess is not the right word. They try to actually impute what the most likely additional missing variants would be, given a given haplotype. But what complete sequence and phasing does, it gives you everything, irrespective of class and irrespective of frequency. So there's nothing more left to figure out from Rob Williams. We know everything from one end of his chromosome to another in terms of the variation. And I have argued, and I mean, this is brainwashing from the days of the Human Genome Project and Maynard Olson, for example, is that we have one simple task as geneticists to do, and that is to completely understand all forms of variation. Once we understand all forms of variation, and I mean just bean counting, understand all forms of variation, we've solved what is essentially a finite problem. Unlike all the people working on transcriptomes, all the people working on proteomics, where that problem is infinite. And by actually even environmental impact is infinite. But we stabilize the equation by actually having complete information. And once we have that, the other associations, I think, will follow.
Yep.
And that's why I think this long read sequencing is so powerful, right? Because it's a lot of work, a lot of money right now to do it. But we can finally close that part of the chapter, right? Understanding every variant that's in your genome, how to genotype it, and so on. That's another story. But making that finite problem tractable, I think, is really important.
I do have more, so anybody interrupt? Bill, I see there, Bob Rooney, BJ, go ahead and interrupt. Anybody with a question?
Or if you're shy, you can type it in the chat. So we were talking, you had mentioned in your slide about the phased haplotypes and finally being able to get an accurate estimate of the rate of de novo mutations, which I thought was interesting. So do you think that will increase the number? And would that reveal some differences in like modern human lineages or among families or populations?
Yeah, I think it's going to change our rate somewhat. I mean, we've done some of the first quads and trios, so from blood. So we have not everything else. Everything I showed today was cell line, right? So there you got issues of somatic mutations and so on that are artifacts of clonal propagation. What we've now started to do is actually families that we've not solved. In our lab, we study autism, and so we're going back to those and we're starting to sequence. The really cool thing is that we actually now can, even if we don't solve the case, we now have data on de novo mutations in regions of the genome that we've never seen before. And I can tell you that the rate calculations are going up. We're discovering, as you might expect, because we're accessing more data, we're discovering more de novos. That's a no-brainer. But the regions that we're characterizing seem to have a more elevated rate, and therefore they're hitting above their weight, which I think is really interesting. It's almost as if, you know, there was a big debate 5 years ago, 6 years ago, of how the clock was too slow. It doesn't make any sense. Some of that, I'm not going to say all of that, some of that has to do with just the fact that we were only ascertaining really, as one of my colleagues would say, self-respecting regions of the human genome, right? That are really important, right? We weren't characterizing the quote unquote junk as well. And now we can do the junk as well, right? So.
And I think you really nicely in that conversation about the regions involved with brain evolution, but also being associated with mutation. I guess that it's even more hard to calculate because our genomes have changed and potentially have some regions that are more prone to different types of mutations versus more ancestral populations.
Yep. And there's certainly some evidence of that, even differences between Neanderthal and humans. Now we're inferring this because we only have short and very short read data for archaics. But I mean, we've now found there's a gene family associated with iron homeostasis that we characterized a few years ago and continue to work on. And that's one that's expanded just in the root of Homo sapiens. So after divergence from Denisova and Neanderthal, which we think is super cool, right? It's the same locus that predisposes us to one of our most common causes of autism, the 16p with 1.2 deletion. I see Rooney had a question there about,
Take Bob's question first, Yeah,
his question, that if I can interpret it there, was it looked like some centromeres had like 2 hypomethylation dips. Yeah, that's a good eye. We've seen this as well. We can't interpret it yet, what that means. And, it could be that, I mean, I guess one way to think about it is that maybe it's so dynamic that it's changing even in the cell at some level. There's some wobble on the epigenetic hypomethylation dip. And maybe that's what we're seeing. The other thing that I don't know if you saw, but it's pretty clear that the hypermethylation regions weren't uniform. I mean, sometimes you would just see that, most of the CPGs were solidly hypermethylated. But in some cases, it looked like there was like half of the sites were hypermethylated and half were not hypermethylated, suggesting that there's heterogeneity at the cellular level going on at some level. So yeah, this is totally Wild West. I don't have an explanation for that. And I will leave it to the next generation like Glennis to provide clarity on that. This is her ball of wax.
For historical reasons, most members in our genetics department, or a lot of them, are neurogeneticists. And I spent a lot of my life counting neurons and measuring brain weights. So I did put a comment in there about causality versus comparative approaches. You made the statement that the Cnvs are enriched in those regions which are human specific or have high rates of evolutionary change in the hominid lineage or the ape lineage or the super primates, whatever division. And I guess I would challenge that and say, if that's the case, I want to see those Cnvs modulating perfectly normal variation between your brain size and my brain size and Megan's and everybody else's. If I don't see it in a causal genetic context, if it's just a comparative gorilla ape, gorilla to orang to macaque to human, I guess there's just so much that's going to be variable there. I do understand the measurement of accelerated regions, so that does add a sort of panache of causality, but it's not enough for me. I need to see it in a segregating population of humans or mice.
Yeah, no, I think I'm on the same. I've always had this discussion. So with people that are, we've come from a comparative perspective, find the regions. And it's interesting that the people that are studying often finding the function of the genes. It's not us that is discovering the function. It's actually neuroscientists that have come, like Frank Pallot, for example, or Wieland Huttner, who've been working on discovering the gene. Then we have met in the middle, basically, is when they realize that the gene that they're characterizing comes from the duplicated regions that are predisposing to the rearrangements. So it's not as if in this case that, we found the comparative analysis and then we looked for the gene. It's really kind of that the genes were being studied separately, and then we came together on this. So that's why a lot of these, as Bob Watterson would say, a lot of these old stories in evolution of the human brain, like circa 2010, were just so stories, because it was like, Rudyard Kipling's Just So, I could tell a story. But these actually have functional data, and were come at, like, as an example, Wieland Huttner, when he was looking for brain genes, that were specific, he basically looked for genes that were from single cell RNA-seq that were expressed, comparing essentially mouse and human, which is a huge genetic distance, looking for a very specific type of gene, a gene that was not found in the other species. And when he identified a whole cadre of these, and then he asked the question of which ones were specific to the radioglial cells, which are the rapidly dividing cells that give rise to the neocortex. And there was one gene, and that gene was RGAP11B. RGAP11B maps to the duplicated sequences that are human-specific that promote the formation of the 15q13 microdeletion, which is the most common cause of epilepsy in the human population, at least due to a CNV. Then what he did, and this is the modeler in him, said this looks like it's the gene important in terms of increasing the number of radioglial cell divisions. So he did a whole plethora of experiments where he showed that if you introduce this into stem cells, it increased proliferation rates and so on. Then he actually took the gene and he made two models, one a marmoset model, and one he made a model of another mammal. And he showed that essentially introduction of this gene actually led to essentially the expansion of the frontal cortex. It didn't make a smarter marmoset. It didn't make a smarter, I don't think he did it in mouse, but it didn't do that. But it did actually have the actual effect, like suggesting that if you have this new function and this new gene introduced into a foreign system, it had the consequential effect.
That's cool.
So I don't take this evidence as lightly. I think it's actually really important to do those types of experiments. And the half a dozen genes that I'm thinking about now, maybe less than half a dozen, maybe 4 genes, they have that level of functional neuroscience that's done behind them. So they're better than the Just So stories from Bob Watterson and Rudyard Kipling. Yeah. They're as close to the real deal as we got right now. Great. I have said from the beginning, actually, and Frank and others, Wieland, have not agreed with me on this. I said, if these genes are so important, I must be able to find humans that are lacking this. And in which case, when I find them, they should have a dramatic deficit in terms of their frontal cortex. So we actually have done screens for this. And have found disruptions associated with kids with developmental disability and microcephaly, but not enough to prove statistical significance yet.
Cool.
I completely agree with you. We got to do, in this case, it has to be both forward and reverse with experimental at the end of the day, I think.
So you've got a tremendous expertise in autism, and one of my questions, what is the status now of CNVs in autism, going back to Wigler, Geschwind, and de novos, and whether, my impression is with the heritability of 0.8, it's pretty damn clear it's just a conventional segregation of some variant. So are there lots of autism specific de novos, or
No, I mean, at maximum, it's about 5% of cases with autism have a de novo CNV. That's probably a maximum. But that's not insignificant. That's a big contribution. A much larger fraction, probably close to 20%, I think that's the most recent calculation, are due to de novo missense and loss of function, just normal point mutation events happening in hundreds of genes. The estimate is 400 to 500 individual genes, each one when mutated in a dominant fashion, can disrupt brain in a sufficient way to lead to an autism phenotype. We've identified, I think, 138 of those genes thus far, just based on analyzing trio data. We put out a paper just a few months ago on this. There's another signal which I think is really interesting. It's inherited mutation. It's private. And what I mean by private is mutations that are gene disruptive and mutations that you don't see if you look at thousands and thousands of other families or even normal genomes. So there are things that arose, and we can show this, that arose in the grandparental or great-grandparental generation. But the really cool thing is that there's the significance and the odds ratio gets even stronger if we require there be 2 or more such mutations. So in other words, it's like digenic, trigenic, ultra rare variants coming together, and the child is unfortunate because it inherited, sometimes it maybe the other event is de novo, one event is inherited, maybe both came, one from one parent, one from the other. Parents are there's nothing abnormal, really, at least from an autistic perspective. But the kid had the misfortune of inheriting both of these loss of function mutations and genes that otherwise are not, what's really surprising to me though, is if we say, okay, go ahead and take all the genes that we know are de novo, 95% of the signal remains. In other words, it's a completely different set of genes that's actually, it's in the same pathways, but they're genes that are not typically the genes that we're picking up as monogenic de novos in these kids.
Yeah,
so I think that's actually very frightening for people that are trying to model the architecture, because that's probably, we estimate in terms of population attributable risk that this may be as important as the CNVs and de novos combined.
That's fascinating. I'm just reminded of the hilarious thing about assortative mating in Silicon Valley and in Seattle. So we'll blame it on you too, of people who have high quantitative competence and marry each other, resulting in this kind of epistasis. That was the Baron-Cohen hypothesis, right, of autism. I don't know where that stands anymore, but I always found that extremely amusing.
Well, what I've learned is you have to be very, very careful about these types of things in terms of what you claim, right? I mean, one of the things that we've seen is sometimes even in these asymptomatic parents, that there's actually endophenotypes that are tracking. You don't see them because they're not going to make, they're highly functional and there's no issues, but when you test them on certain scores, like social responsiveness and so on, they often show kind of extremes that would be consistent with moving toward this autism spectrum, but clearly wouldn't make diagnosis.
Yeah. Cool. So I got a huge kick out of your saying we shouldn't bother to teach cytogenetics to M1 students, because I just had to teach genetics to the M1s, and I taught 0 lectures on advanced genomics, or maybe 1. And then there were a series of 5 lectures on cytogenetics. I was going, oh my God, we're teaching our poor M1 students genetics circa 1955 or 60.
Well, I mean, I teach a class in the winter quarter with Mary Claire King, and it was about 3 or 4 years ago. And I think for like a decade, she had a section, an entire 2 weeks on linkage analysis as part of her training. And I mean, that was her lectures, and I didn't mess with it because she knows that literature well. But then she finally came to me one day. She goes, "I think I should stop teaching linkage. It's not really that relevant anymore." I mean, it's important, right? I mean, it's really important thought and process in terms of all odds calculations and so on, and it's fantastic, but it's really not relevant, right? I wouldn't say that about cyto. I still find cyto useful in very specific cases, right? So I still think you could argue that learning some cyto is not a bad thing.
Yeah, because I mean, because we had Ernestine, a graduate student, ask questions about, like developing countries and different technologies. So, I mean, if you at least knew where to look, then you could perhaps leverage some of those new, I forgot the name of the machine that does the like real-time targeted sequencing. So
Oh, you mean like the Oxford Nanopore? Yeah.
Yeah, sorry, ONP. Yeah.
ONT. Yep.
So
Oh, definitely.
So yeah, I mean, that would be a really simple starting point.
One big question that's relevant to us here is, where do you think it's going to go with newborn genetic screening, newborn genomic screening in Washington State over the next 5 years? Is anybody agitating to sequence every baby in Washington?
Yes.
I'm gonna
actually,
stop recording. Okay.
actually, the Danny Miller, he's basically doing exactly this. He's starting a project